All right, so why don't we go ahead and get started? So thanks everyone for coming out today, hopefully before many of you leave for Thanksgiving. So it's my great pleasure to introduce Kristin Koutmou from University of Michigan, where she's the Seihan Egi Assistant Professor of Chemistry, as well as the Associate Director of the Graduate Program in Chemical Biology. So Kristin, she got her undergrad degree from UC Denver and then went straight into the RNA world with graduate work at Michigan with Carol Fierke, where she worked on RNase-P. Then after that, she worked for Rachel Green at Johns Hopkins, where she began studying translation and kind of the mechanics and inner workings of that. And that's something that's carried forward, as you probably can tell from the slide here, to her present day work in her own lab at Michigan. So Sarah has done incredibly well, and she's won a number of awards, including the Scilog Fellow Award, a Cottrell Scholar Award, and an NSF Career Award. And her research I find to be really fantastic because it kind of bridges this incredibly important problem in biology about post-transcriptional RNA modifications at multiple levels. So how they're made, so the enzymes that put them in, how many of them there are, which is an incredibly important and poorly understood question in biology, and something which I think we'll hear about today with some mass spec work. And then finally, how these impact gene expression and translational mechanisms in general. And so I want to make one final thank you before I introduce Kristin, and that's thanks to Thermo Fisher. So we have a number of representatives here, including some alumni of the Biochem department from Mike Cox's lab. And so they're sponsoring today's seminar, so I want to thank them for that. So with that, I'll turn things over to Kristin, and thank you for visiting Madison.
Thank you so much for hosting me, Aaron. It's been a fantastic day here. I have really enjoyed learning about all of the fabulous science happening here at Madison and seeing old friends and getting to meet some new ones. So thank you also to everybody I've interacted with today. I've had an absolutely fantastic visit. Cells face the daunting task of having to maintain the right number of proteins in the right place at the right time. One general strategy they use to do this is to add chemical modifications to all three of the major biomolecules in the central dogma in order to control their structure, function, and stability. So when we take a biochemistry class, we tend to talk a lot about the sorts of modifications that get incorporated into DNA, things like methylations that are important for regulating gene expression in the epitranscriptome, and in proteins, things like phosphorylations or ubiquitinations that are important for controlling protein activity and localization and stability. But it turns out that RNAs are also highly modified, with over 150 different varieties of modifications having been reported in non-coding RNAs. These modifications have been known to exist since sort of the dawn of RNA research in the 1950s. And until very recently, these modifications were thought to be solely the purview of non-coding RNAs, which is just a catch-all term for every RNA in the cell that is not an mRNA template for protein synthesis. However, the last 10 years have really flipped this dogma on its head with the discovery of roughly 15 different types of modifications in protein-encoding messenger RNAs. And the discovery of these modifications has largely been made possible by the advent of deep sequencing technologies that have allowed scientists to develop methods to go in and map the location of individual modifications transcriptome-wide to every RNA in a cell. And as they've done this, they've seen, guess what? They're not only in tRNAs and ribosomal RNAs, but they're in messenger RNAs as well. The idea that messenger RNAs can contain modifications has really exploded and launched an entire new area of research because the imagination of scientists worldwide has been captured by the idea that these modifications can be incorporated into RNAs in order to impact every step in their life cycle following transcription. So you can imagine how a chemical modification might alter the stability and dynamics of an RNA, how it can interact with a protein, its stability, its splicing and maturation, and even how the ribosome itself might be decoding an mRNA message. In line with all of these possible functions for mRNA modifications, the two most well-studied modifications in mRNA, N6-methyladenosine, also known as M6A, and inosine, have been linked to a myriad of diseases, including cancers, neurological disorders, and cardiovascular diseases, as well as to a variety of cellular stresses. At this point, I like to say that you can pick any disease that it is you would like to study or are interested in, type in plus M6A into Google, and you will have found someone that has mapped the location of M6A in those disease states and seen that it redistributes. Sequencing studies, like the ones I've just described, have laid a really important foundation for the mRNA modification field, demonstrating that modifications can in fact be incorporated into mRNAs and that they can redistribute in response to different cellular states, environmental conditions, or diseases. These studies have given us a beautiful bird's eye view of where modifications can be in the transcriptome and how these modifications can redistribute. However, large correlative studies can't really tell us what any individual modification actually does. And so the mRNA field moving forward, sort of the next big step for scientists in this area, is going to be moving beyond simply mapping modifications to trying to develop a quantitative foundation for understanding how they actually change interactions between an mRNA and components of the cellular machinery. And that's ultimately what the overarching goal of the work in my lab is to do. It's to move beyond these beautiful landscape maps we have down to what I call the Google street level view, where we zoom in on individual modifications. And then my computer asks me to update. Just a second to quit out of that. Okay, where we zoom in on individual modifications and ask how they one, are incorporated, how much of them is there, and how they ultimately impact gene expression. We get at this question in sort of 4 general ways in my lab. The first way is that we develop and implement mass spectrometry tools in order to discover new RNA modifications, quantify the amount of modifications that are present, and we're actually working on developing direct sequencing of RNAs by mass spectrometry as well in order to see in a very precise way where modifications are incorporated. Using the technologies that we have developed so far, we've discovered 6 new modifications in yeast. This may sound like a small number, but when I began working in this field roughly 5.5 years ago, I think there were 4 reported RNA modifications, suggesting that the platforms that we've developed are actually quite a robust way to do discovery. And in addition to identifying new modifications, we've also identified the enzymes that put those modifications in. So RNA modifying enzymes are most famous for being able to target tRNAs and ribosomal RNAs, and most of the enzymes that put modifications into mRNAs are the same enzymes that do so into these non-coding RNAs. So the dogma in the field for these particular classes of enzymes have been that they recognize RNA secondary structure. So if you think back to your biochemistry class and you just kind of imagine in your head what a tRNA structure looks like and what you would imagine an mRNA structure to look like, that might lead you, just like my lab, to want to revisit this hypothesis that the enzymes are only targeting RNA secondary structure. And my lab is working to try to understand how these really long-studied enzymes are targeting this entire new class of RNAs, mRNAs. And we've done some early work demonstrating that for at least some of these proteins, the actual inherent preferences of the proteins for particular structures or sequences matters much less than their accessibility to those two particular RNA targets, and that RNA localization and protein localization actually matter a lot for determining which mRNAs are ultimately going to be selected for modification. The largest portion of work in my lab is really focusing on the molecular level consequences of mRNA modifications. In particular, we focus on consequences on translation. This may sound like an easy question to ask, but it's actually not because of this problem right over here. Those enzymes that put in modifications into mRNAs that we're interested in studying also put them into tRNAs and ribosomal RNAs. So if you just knock out these modifying enzymes and look in a cell like at a reporter and say, oh, you know, without that enzyme there, we make less protein, you don't know why. You don't know if that's because there's a difference in the mRNA modification status, the tRNA modification status, or the ribosomal RNA modification status. This makes asking questions about what happens when the ribosome encounters modifications actually pretty tricky. And my lab gets around this problem by using a totally reconstituted in vitro translation system where we have all of the components necessary for translation purified. We add them back together in discrete amounts and do transient kinetics with the ribosome. And we can look at which individual step in the translation pathway is being impacted by individual modifications. Some of the work I'm going to be talking about today speaks to this. In particular, I'm excited to share with you about some stories that we have in lab where we've discovered that these modifications generally change both the speed and accuracy of the ribosome in order to impact protein production. Lastly, fortuitously or not fortuitously, right before the pandemic, my lab got very interested in RNA viruses, which it turns out also contain a lot of RNA modifications. We're interested in asking the same sorts of questions we are about the molecular level consequences of modifications in mRNAs. Where we are using mass spectrometry tools that we've developed in order to identify and quantify modifications in RNA viruses. And we have several recombinant RNA viral systems up and running where we have RNA-dependent RNA polymerases purified, and we're taking a look at how the modification of viral RNA genomes ultimately impacts the fidelity and rate of those enzymes. And suffice to say, it does. The focus of my talk today is really going to be on these two portions of work in my lab. I'm going to start off talking briefly a little bit about some of our mass spec work for doing modification discovery, and I'm then going to move on to, to speaking about the consequences of RNA modifications in translation. So we've known about the inclusion of RNA modifications into non-coding RNAs for a really, really long time. I think pseudouridine was first discovered in tRNAs back in the late 50s. And the reason that we've known so much about the modification of non-coding RNAs is in large part technical. It's because non-coding RNAs make up most of the RNA in the cell. And if you have tools of relatively limited sensitivity, these are the RNAs that are the easiest to study. They're the ones you're most likely to be able to see modifications in. Protein coding mRNAs are only really 1% to 2% of the total RNA in a cell. And we've known, many of you may be familiar with, in fact, the 5' methyl G cap that's put on the end of all messenger RNAs. But for the most part, we didn't have any idea about what else might be in there, and that's largely due to a detection problem. Now that we have these deep sequencing technologies that can allow us to ask questions about what's going on in the protein coding mRNA space, I think we have some grand challenges for mRNA modification discovery and mapping, figuring out where those modifications are and what it is they might be doing. So the grand challenges for mRNA modification discovery, which I think should still be a real thing, a real field. I don't think we've discovered all the mRNA modifications because there's 150 different varieties possible in non-coding RNAs. It's hard for me to imagine that nature would limit itself to less than 10% of those in mRNA. So I think this is a reasonable thing to continue going after. The first important question when trying to study mRNA modifications in a sample is simply, what is there to begin with? The next major question to ask is, now that we know what's there, where are those modifications? What types of RNAs are they in? Are they in tRNAs, ribosomal RNAs, mRNAs? What sorts of RNAs are they in? And the third important prong is how much of each of those RNAs is actually modified. So the field has really used very robustly sequencing studies in order to ask where modifications are, right? To make these really elegant maps of where modifications can be in a transcriptome. But using solely sequencing studies to do this can be quite limiting because these sequencing studies are incredibly laborious to develop. It's actually pretty hard to ask what's there in a sample. They can allow you to look at one modification at a time. And when you're thinking that there's a possibility of 150 different ones, that's a pretty limited question. They also are not quantitative. They can't tell you how much of any given modification is there. So when I started my lab as a chemical biologist, being a newbie to the field, I came in and said, well, I know that protein post-translational modifications are pretty intellectually analogous to these post-transcriptional modifications that I'm interested in looking at. Why not think about using some of the long-standing technologies that people have put in place for studying PTMs to also look for RNA modifications? This was sort of a naive thing to do. It turned out to be way harder than I expected, but we've had some success in that space, and that's what I'm going to share with you about today. So we decided to use mass spectrometry initially in order to hunt for new RNA modifications because, again, it seemed to me that sequencing-based approaches were not the best way to do discovery, that we needed a more unbiased way to look for modifications. And as we're trying to figure out which modifications are most biologically relevant, we also needed a method to be able to quantify the amount of modifications that were present in a given sample. And mass spectrometry is really an ideal way to do that. So what I have shown here are the two main people who were involved in this work. I will freely admit to you that I am not an analytical chemist by training. However, I'm in a fantastic department full of lots of wonderful analytical chemists. So I collaborate with Dr. Bob Kennedy, who is a mass spectrometrist and is quite famous for some proteomic and metabolomic works. And he and I share a student, Joshua Jones, who took on all of my harebrained ideas and was able to make them work. So Josh developed an LC-MS, so liquid chromatography followed by mass spec assay to look for new modifications in mRNAs. In this assay, he has not reinvented the wheel. He has taken what is more or less a standard workflow and improved it dramatically in order to allow us to look at low abundant mRNA modifications. So he purifies out mRNA from the cell, digests it down into nucleosides, runs a Hillock column, so performs liquid chromatography where he separates out all of the nucleosides that are present in his sample, and then he runs MS and does multiple reaction monitoring in order to detect all of the nucleosides that are present in a sample in parallel and quantify them because he has run a series of standards up front so he can tell how much of everything he has. This is the way that non-coding RNA modification discovery was done largely by Jim McCluskey for many, many years. However, the standard form of this type of assay was not sensitive enough in order to be able to detect the low abundant mRNA modifications we were looking at. So Josh really improved this platform in two different ways. The first way was to simply highly purify mRNA in a way that was believable, that we weren't looking at contaminating tRNA signals. And to have large, and come up with schemes for having large amounts of mRNA. The second really key innovation was to improve the chromatography in front of the mass spec. And this step in particular drastically increased the sensitivity of our assay. So why do we have to do chromatography up front? Why is making a chromatography innovation an important thing to have done in this space? Well, this isn't a normal question to ask because, modifications have different molecular weights. Why can't we just shoot them onto the mass spec and look at them all individually? The reason that chromatography is so important in this system is because these modifications are present in very low abundance relative to canonical bases. So for example, if you were to take total S. cerevisiae RNA, digest everything down, we know there's about 30 different modifications that exist in S. cerevisiae, and you were just to shoot them on the mass spec, you would see great intensities for cytidine, uridine, guanosine, and adenosine, and then you could see little tiny peaks for a couple of the RNA modifications. You basically just can't see them because they're totally swamped out by the canonical nucleosides. And so when you take these and then you take this sample and you directly inject it in the mass spec, any of the signals from these little guys down here are being totally suppressed by the canonical signals, and you just don't see anything at all. You get large amounts of ion suppression. So what Josh did was he developed some hillock chromatography that allowed him to look and separate out 50 different modified RNA nucleosides. And by doing this, he added 15 more modifications, the ability to detect 15 more modifications quantitatively than previously existed. He saw no ion suppression of co-eluting analytes. He increased the analyte dwell time and decreased the flow rate for better ionization. Together, this allows us to now look at 50 different ribonucleosides in parallel in a single assay, and to be able to detect them quantitatively, because he creates curves for each of those 50 nucleosides. These 50 nucleosides, because they're separated out so well, have quite a wide linear dynamic range over 4 orders of magnitude, and he's able to detect to have the lowest detection limit for single modifications at 3 attomoles per 0.6 picomolar, which is actually quite sensitive. In fact, just this chromatography innovation alone increased the sensitivity of our assay for 15 modifications, giving us the highest sensitivity assay for being able to detect these modifications, which is key for being able to study mRNA modifications, which are much less abundant than their canonical nucleosides. Because if you think about how long an mRNA is, if you have one modified site, that's very rare relative to the total non-modified RNAs. Okay, so now he's developed this incredibly sensitive system, and he went on and he wanted to do the next hardest thing, which was to not just look at all of the modifications on non-coding RNAs, but to go in and try to use this to discover new modifications in protein-coding mRNAs. This is, again, a very difficult thing to do because very few of the RNAs in a cell are modified, and these modifications exist at low level, in contrast to, say, tRNA modifications, of which there are many on a single RNA molecule. So the first thing that Josh had to be able to do was to purify out these protein-coding mRNAs. And this is extremely important to do well because if you have any non-coding RNA signal around, you're going to be having your your LC-MS/MS assay is contaminated by these highly modified species, and you do not want that. But it turns out it's actually really hard to purify something that there's only 1% of in a satisfying way. And that really is one of the things that's limited the utility of these sorts of screening mass spec assays for mRNA. So Josh developed a very robust mRNA purification pipeline in which he purified total mRNA from yeast cells, first performing a small RNA depletion, followed by 2 poly-A pull-downs, followed by an rRNA depletion, followed by 4 orthogonal quality control steps, some of which I will walk you through here. The first quality control step was simply a bioanalyzer, which is a capillary electrophoresis that separates out RNAs based on size. And you can see that starting in the total RNA, he had very little mRNA signal. You can barely detect it there. He mostly saw tRNAs and ribosomal RNAs. Following small RNA depletion, he had many fewer tRNAs. Following poly-A enrichment, he got rid of all the tRNAs, and now you see a large mRNA signal. And then following that rRNA depletion, the rRNA is gone as well, and you really only see things of an mRNA size. He also did RNA-seq, and you can see that in total in the total RNA samples, he had primarily non-coding RNA and very little mRNA. And then in the purified mRNA samples, he saw basically no signal from non-coding RNA species. However, you might say to me, well, Kristin, RNA-seq isn't very good at looking at tRNAs, and that's the contaminating species you should be most worried about. Never fear. We were able to use our mass spec assay in order to ask that question. So one of the benefits of looking at 50 different modified nucleosides in parallel is that you have a number of negative controls internally that you're already using for every sample you run. So we actually didn't detect almost all of the tRNA modifications that we had the capability of quantifying in our mRNA samples. So I have examples of those shown here where we don't see, for example, dihydrouridine, M3U, T6A, I6A, or some fancy modifications found on a handful of RNAs. Dihydrouridine in particular is important to note because there's 1 to 2 dihydrouridines per every tRNA. So if we don't detect that in our sample, that means we do not have tRNAs in our sample. We then use the limit of detection for dihydrouridine in order to measure the theoretical maximum amount of tRNA contamination we could have to be 0.002%. I think that this number doesn't mean very much. The thing that means a lot to me is that whatever tRNA modifications are there, they are below our limit of detection, so they're not going to be a problem. We used this assay then to look at how the levels of different mRNA modifications change in cells in response to stress, which actually had not been done quantitatively. People had seen that modifications redistribute, but nobody had gone in and looked to see just quantitatively how their levels fluctuate in response to different stress conditions. And the take-home message from this slide, where we looked at mRNAs purified from a yeast grown in YPD, H2O2, so oxidative stress, heat shock, or glucose starvation, is that they do change. So modification levels do change in response to stress. We also took our yeast mRNAs, and there were a handful, not very many, different modifications that we saw in our yeast mRNAs that hadn't been reported in mRNAs before. We've seen 6 of them so far. I have 4 of them shown here, where we now detect M1 guanosine, M2 guanosine, M22 guanosine, and M5U, also known as ribothiamidine, in mRNAs. Consequently, we found the enzymes that insert these modifications. These are all tRNA modifying enzymes. And we've done subsequent translation work on these modifications, looking at how they can impact ribosome action. And I'm going to talk about that here in a little bit. So in sum, for this mass spec portion of the talk, we have developed a new pipeline for studying mRNA modifications by LC-MS/MS. This pipeline has been vastly improved over what was available before in two ways. One, through rigorous mRNA purification, so we can have confidence in what it is we're looking at. And two, through new chromatography that has allowed us to vastly increase the sensitivity of our assays. And these sorts of assays have allowed us to look at things like not only new mRNA modifications, but also look at how the tRNA modification landscapes change in response to stress, look at how modification landscapes in RNA viruses change, and a variety of other questions that I'm not going to list here for you today. Okay, so these mass spec sorts of studies really play in then to all of the areas and touch on some of all of the areas that my lab studies. In particular, these RNA modifying enzymes. You know, we're discovering new RNA modifying enzymes using these mass spec assays, opening up new avenues for us to study. And we're also discovering new modifications that are present in mRNAs. And that's what I'm going to talk a little bit about now, is looking at the molecular level consequences of modifications on translation by the ribosome, including one of the modifications that we discovered in that previous study, 5-methyluridine. So when my lab started, I was interested in first looking at the molecular level consequences of uridine modifications on mRNAs because of the discovery by Schrager Schwartz and Wendy Gilbert of pseudouridine in mRNAs. I thought pseudouridine seemed like an interesting target to study as someone that's interested in translation because it's very abundant. It's actually, if you do mass spec assays, as abundant as the most famous M6A modification in mRNAs. It's also primarily found in the coding sequences of mRNAs, which is the region of the mRNAs that the ribosome reads and actually translates, suggesting to us that this is going to be a sequence that the ribosome is going to be encountering, or a modification the ribosome is going to be encountering very regularly in cells. And these modification is also enriched at RNA binding protein sites, suggesting to us that it might be physiologically important. There was also some data indicating that this modification is incorporated at high occupancy. This is an important caveat because the sequencing studies that are available out there can tell us whether or not a modification exists, but they generally do not tell us if it's there 1% of the time or 100% of the time. And when you're trying to figure out which modification sites are most physiologically relevant, this is really important information to know. And the idea that pseudouridine can be incorporated at up to 90% of the time at certain mRNA sites suggested to us that it might be having a biological function. I often get questions about this from people in the RNA field who say, well, you know, in ribosomes and tRNAs they're there 100% of the time. If they're only there 50% of the time in mRNAs, do you really think they're doing anything? And I just want to remind everyone that protein post-translational modifications, which I think we probably all agree do something important, are also in proteins substoichiometrically, and just because they're not there all of the time doesn't mean they're not doing something important. And in fact, the levels of modifications we see in mRNAs are quite analogous to that for things like protein acetylation, phosphorylation, and glycosylation, which are all generally substoichiometric as well. The other reason I was very interested in pseudouridine is because the incorporation of pseudouridine varies with stress, which our mass spectrometry assays had shown, as well as some sequencing studies, as well as with different stages of development, again, suggesting that it might be important or have important consequences on gene expression under different cellular conditions. So we started off looking at pseudouridine. We also looked at two other mRNA modifications. The first being M5-methyluridine, which we had discovered is present in mRNAs by our mass spectrometry studies. In the meantime, a couple of other groups have also found M5U in RNAs using sequencing studies, which really, of course, gives us more confidence in our mass spectrometry abilities. And then we were also interested in N1-methylpseudouridine, and we picked to study this one because it is a modification that many of us here may have actually had in our bodies because it is found and used robustly in all of the current mRNA vaccine platforms. So before I go on, I always like to give a brief shout out to basic science, because when we started this particular project, although I would like to be able to tell a gorgeous story about how we were just following up on observations we'd made in cells and some really cool therapeutic technologies that were emerging. When we started looking at M5U and M1 methyl pseudo-U, I really just begged my students to do these as molecular level probes of pseudouridine. I was interested to see if this nitrogen on pseudouridine was having any effect, and then just using M5U as a control for N1 methyl pseudouridine, and I had to beg my students to do these experiments because I thought they're not biologically relevant. They don't matter, they're not important, but they kindly did them, and then that data has ended up being really, really interesting because now we know that M5U is in mRNAs, and COVID vaccines happen, and so there's all of a sudden a lot of interest in what happens when the ribosome encounters N1 methyl pseudouridine. So yay for basic science. You never know when some fundamental question that you ask is going to end up being really important. So the work I'm going to present to you on uridine modifications was done by some fantastic members of my lab, some of whom are still in my lab and some of whom have graduated. Mehmet now runs his own lab at the University of Istanbul, and I think everybody up here has graduated except for Josh. And I'd also like to give a shout out to my wonderful lab staff, including Taslima Khan and Dr. Dan Eiler. So as I mentioned at the beginning of this talk, asking what happens when the ribosome encounters an mRNA modification is not such an easy question, and we get around that by using a reconstituted system. So the easiest question to ask to begin with is to simply say, well, what happens to the speed of the ribosome as it's translating along when it hits one of these RNA modifications? We've done this by purifying all of the components necessary for translation and forming ribosome complexes with an S35 radio-labeled methionine tRNA in the ribosome P site in what we call an initiation complex, and a phenylalanine codon containing either unmodified or singly modified phenylalanine codons in the A site. We then add all of the downstream factors necessary for translation to happen and follow the formation of a dipeptide bond by electrophoretic TLC. When we do this, we can look at the kinetics of translation elongation, and we see that the insertion of M5U into our sequence slows the ribosome down by about twofold, but in a context-dependent manner, only when that modification is present at the second position. For pseudouridine, we see a very similar trend where the ribosome is slowed down. However, now this is more uniform across all three positions in the codon. Follow-up studies where we are starting to try to deconvolute the 7 kinetically definable steps in translation elongation to figure out which ones in particular are being perturbed by this modification demonstrate that the inclusion of pseudouridine slows a key step in the translation sort of quality control pathway, where when tRNAs bind, their EFTU hydrolyzes GTP before it leaves the ribosome and can go off, and this particular step is slowed down, again suggesting that pseudouridine should slow the ribosome. There was a follow-up study of our work that showed that what we see in vitro actually does happen in cells as well. So studies by Levi and Arava in 2021 looked at one particular enzyme that inserts pseudouridine into mRNAs, pseudouridine synthase 6, or Pus6. Pus6 inserts pseudouridines not only into mRNAs in yeast cells, but also into elongator tRNA methionine. When they knocked out Pus6, they saw that the overall levels of protein in the cell went down. This makes a lot of sense because they've mucked with one of the key elongator tRNAs, so less protein is being made because this tRNA isn't in good shape. That's fine, that's interesting, but it wasn't what was interesting to me about this paper. What was interesting to me about this paper was the fact that even though total protein levels were down because there was a problem with this tRNA, the levels of proteins translated on mRNAs that contained a pseudouridine inserted by pus6 were increased, suggesting that when that pseudouridine is there, you're making less protein, consistent with the ribosome slowing down on these transcripts. We also worked with Dr. Yuri Polinikov at the University of Illinois at Chicago to solve a crystal structure of a 70S ribosome on a unmodified and pseudouridinylated mRNA codon. These crystal structures revealed that the base pairing between pseudo-U and the mRNA looked pretty normal. And the tRNA looked pretty normal, but the other end of the tRNA, the sort of business end of the tRNA, that 3' CCA that brings in the amino acid into the ribosome to react, was disordered for tRNAs that were bound to a pseudo-U containing codon. This is the sort of thing that crystallographers typically see with the ribosome when there's a problem with accommodation, which is a key step in translation elongation. In addition to pseudouridine, we also looked at M1 pseudouridine, which did not seem to really have much of an impact on ribosome speed. Okay, everything I've told you so far suggests that these uridine modifications can slow the ribosome. And that's interesting. It can impact how much protein gets made. It could perhaps, allow time for co-translational processes to happen. However, secretly, the question I was really interested in when I started on this project was whether or not these modifications might influence which tRNA the ribosome selects. The reason that I thought this might be a good question to ask is because pseudouridine has been studied for decades in the context of tRNAs, where it's been shown to make base pairs not only with adenosine, but with other nucleosides as well, suggesting the possibility that this might cause the ribosome to choose a different tRNA to base pair with an mRNA containing a pseudouridine based on these alternative base pairing possibilities. So we went ahead and investigated this possibility, again making use of our totally reconstituted in vitro translation system. This time we have our ribosome complexes poised and ready to react, but instead of providing those complexes with a single tRNA, that phenylalanine tRNA, which is supposed to go in, we provided it with all of the tRNAs that are present in the cell and just looked in an unbiased manner to see what gets made. Do we only make the expected MET-Phe dipeptide, or do we also have alternative amino acid selection and make a bunch of other stuff? Of course, we made lots of other stuff, or I wouldn't be here talking to you about it. Shown here on the left is an electrophoretic TLC with the dipeptide products from this reaction. You can see we make quite a bit of the expected MET-Phe dipeptide, the canonical correct peptide. However, we also made a lot of other dipeptides. Follow-up studies showed that we were able to misincorporate a number of near cognates, including isoleucine, leucine, and serine. We saw very similar behavior with M1 pseudouridine programmed in the A site, where when we fed the ribosome with this phenylalanine codon, a variety of purified tRNAs, we saw much more misincorporation on the M1 pseudouridinated codon than we did on the unmodified codon. We, of course, then went on and verified this with further kinetics, because I'm a mechanistic enzymologist, and if I get to measure rate constants, I just can't help myself. So we went ahead and looked at the kinetics of misincorporation on phenylalanine codons with a variety of tRNAs, including tRNA lysine, serine, and leucine. This is just like the assays I ran at the outset, only now instead of looking for MET-Phe dipeptide production, we're looking at the production of a miscoded erroneous dipeptide. When we do this with serine, we see absolutely no change in the kinetics of misincorporation. So having an M1 pseudouridine or pseudouridine does not seem to change the propensity of the ribosome to put in a serine on a phenylalanine codon. Leucine was generally slowed by the addition of these modifications into a phenylalanine codon by 0 to 3-fold, depending on where that modification was within the codon. And isoleucine was even funkier, where it could either increase where M1 pseudouridine and pseudouridine could either increase how fast isoleucine got put in by 3-fold or decrease it by up to 10-fold, just depending on where that modification was in the codon, suggesting that the effects we should expect to see are highly context dependent. And we see more or less the exact same sort of thing with other modifications as well, including M5U, which increases the rate constant for isoleucine misincorporation by 2-fold, but again, only when it's in the 3rd position of the codon. Pseudouridine looks the exact same as M1 pseudouridine, where it increases the propensity for miscoding when it's in the 1st position of the codon and decreases it when it's at the 2nd. And again, isoleucine looked like that as well. The funkiness of these two patterns was strikingly similar for these two modifications, suggesting to us that it has something to do with alterations in the base pairing of the normal Watson-Crick face of the nucleoside. So when you get observations like this in a test tube, in a reductionist bacterial system, it's always important to ask yourself, is what I'm seeing in my test tube actually true, or is it just an artifact? Can it actually also happen and hold up in cells? So we asked this question in human cells in collaboration with Dr. Bejoyta Roy when she was at NEB. She's now at Moderna Therapeutics. In Dr. Roy's system, she in vitro transcribes luciferase reporter mRNAs either without modifications or with fully substituted pseudouridine or M1 pseudouridine nucleosides. She transfected these RNAs into HEC293 cells, purified out the full-length protein products, and then did an unbiased mass spectrometry assay looking at every position in the peptide to see what amino acid was being incorporated. When she did this, she saw the exact same thing at pretty much the same level we had been seeing in vitro. So what I have shown here is the peptide, luciferase peptide that flew the best and for which we had the best sequencing depth. Each of the codons that had no U, where we did not expect to see substitutions, are not bolded. Those that contained a U that was substituted to a pseudouridine or M1 pseudouridine are in bold. And codons where we observed substitutions are shown in bold in red. And the thing I really want you to take home from this is that not every single position that could have a substitution does. So this is happening in some sort of context-dependent manner, and that not every position where there was a substitution were those substitutions just going in willy-nilly. They seem to be typically going in with near cognate tRNAs. The other thing to note is that we saw very low levels of amino acid misincorporation in peptides generated from unmodified mRNAs. Less than 0.05% of those peptides had substitutions, and they were only seen on valine codons. This is in contrast to the levels of substitution we saw in peptides generated from modified mRNAs, which were around 1% and on a much wider variety of codons. More recently, let's say within the last week, Chuan He recently published that this can also happen on endogenous mRNAs, where he and his lab developed some new mapping methods that were a little bit more quantitative and more sensitive and allowed them to see that there are actually a number of mRNAs that contain pseudouridine at stop codons. They then went into the endogenous system using antibodies in mice and saw that these codons promoted stop codon read-through with mass spec data indicating that this read-through did not come from a frame shift, but from actually a tRNA binding and interacting with these pseudouridine-containing codons. And when they looked at 10 different transcripts that this happened with, they saw read-through or miscoding akin to what we saw in only half of them. So this indicates that this doesn't happen all the time, which is actually what I would expect. And they also saw this happen in a very tissue-dependent manner. So what I have shown here are two examples from the paper where here's an example of one mRNA that contains a pseudo-U at its stop codon, and nothing happens at all when they looked at that mRNA. However, for Selenoff, they saw that in kidney cells or in kidney tissues, they actually got extended peptide products that were the product of read-through when that pseudouridine containing mRNA was there. But this was not generally true in all tissue types, but seemed to be very tissue and context dependent. So we are now very curious about this context dependence. What is going on? It's very clear that it's context dependent in vitro. It's clear it's context dependent in HEC293 cells and now in mice organisms as well. So we now are taking a fundamental approach to try to understand what might be driving some of that context dependence. We started off by doing some thermal melting reactions to see if there is a change in base pairing stability depending on the context of the RNA. So we started off by looking at self-annealing duplexes with U substituted for M1 pseudo-U or pseudo-U. And what we see is that we're able to measure the melting temperatures that are near what you would predict the melting temperatures to be for these short oligos. And these temperatures are generally increased for this oligo by the inclusion of pseudo-U or M1 pseudo-U. This was not the case when we simply changed that oligonucleotide by a single nucleoside, suggesting where the effects were now much smaller and there was really no effect for M1 pseudo-U, suggesting that the sequence context surrounding that modification really does matter for base pairing stability. And base pairing stability is, of course, going to be an important driver of which tRNAs get into the ribosome, because that relies so much on Watson-Crick interactions between a tRNA and an mRNA codon. Now that's really simplistic. In the context of the ribosome, we all know that there's more going on than simply 3 base pairs in order to drive the interaction between a tRNA and an mRNA. And I just have that illustrated here by a crystal structure where we have an incoming tRNA and an mRNA codon. None of the proteins in the ribosome are even displayed here. And you can tell that there's more complex things going on than only base pairing interactions between tRNAs and the mRNA. So to start to ask questions about these interactions in a more sophisticated way, we worked with Dr. Aaron Frank when he was at the University of Michigan. He's now at Rackus Therapeutics in order to do some molecular dynamics simulations of base pairing between modified mRNA codons or modified phenylalanine mRNA codons and isoleucine tRNAs, looking at those interactions that aren't supposed to happen. I will have to say, and he did this in the context of the ribosome, so he used the crystal structure that we'd collected with Yuri Polynikov as a starting place for his really sophisticated molecular dynamics interactions. So he was looking at these interactions in the context of the ribosomal A site. And I will say that I did not think we were going to see much by these. I was a little bit skeptical, but they completely recapitulated our biochemical data in a way that was really satisfying to me as someone who's a chemist at heart. So Aaron's calculations showed, just like very much in line with what we measured in vitro, that when a pseudouridine was substituted into an mRNA codon in his calculations, he saw higher levels of interaction, better base pairing energies when the substitution was at the first position, where we see more isoleucine misincorporation, and weaker interactions at that second position, where we saw much less isoleucine misincorporation, suggesting really that fundamental principles, fundamental properties of these bases and base pairing are largely what's actually driving this context dependence. And he saw the exact same thing with M1 pseudo-U. Okay. So now let's talk very briefly in my last 2 slides about the possible biological consequences of these things I've been telling you about. So modifications are in mRNAs. There's a lot of data from not only myself, but from others in the field suggesting that these modifications generally slow the ribosome down. And it's easy for me to rationalize why this might be important for regulating, or even maybe not for regulating, but as a consequence on gene expression, right? If you're slowing the ribosome down enough, you can be making less protein. If you slow the ribosome down too much, you can all of a sudden get ribosome collisions where now you trash not only the protein but the mRNA as well. You can imagine that there are a lot of co-translational things that happen in cells that need to be timed, things like chaperones coming and interacting with the ribosome, and it might be useful to pause the ribosome transiently to do that. And that all made a lot of sense to me. But our observations that these modifications are all over in biology and causing low levels of miscoding was much more of a mystery. And so that led us to sort of go on and think, well, what's actually going on? What could the biological consequence or benefit of keeping low levels of miscoding around be? And what we've ultimately come to is a hypothesis that these modifications are likely important under stress. And our hypothesis is that they might provide the cells a way to transiently increase the diversity of the proteome without having to permanently modify their genome. So why do we think this might be the case? Well, we know that low levels of miscoding, meaning the misincorporation of amino acids by the ribosome, can enhance cellular fitness in microbial systems under stress. This is a well-documented phenomenon under a wide variety of stresses. We know that pseudouridine in mRNAs, its levels are increased significantly under stress by like 20 to 50-fold, depending on the system that you're looking at. And that these pseudouridines redistribute also with cellular stresses in eukaryotes. We also know that there's a negative correlation between yeast fitness and robustness. And what I mean by this is basically that yeast that are too faithful at doing translation, that do their job perfect every time, do not tolerate stress well. And they don't survive so well in the environment because they have no wiggle room. So that's what we're thinking about for biological consequences. I think it's also important to note that there can be consequences for mRNA-based technologies because the modifications I've been talking about here are the exact modifications that are being used in all of the current mRNA vaccine and therapeutic platforms. And I'd like to talk for 2 seconds about when and why I think that might matter. So for the most part, I think it doesn't matter for mRNA-based technologies. I believe in the vaccines. I think things are being made right. I'm not worried about any of that. So I want to be very clear in making this statement. However, we do have to acknowledge that on unmodified mRNAs, given the error rates that we've seen both in vitro and in HEK cells, we would really expect to see maybe one misincorporation in a small pool of peptides, to whereas off the fully modified mRNAs, we would expect to see probably something along the order of one misincorporation per peptide. However, those are likely to be distributed sort of randomly throughout the peptide, so I wouldn't expect that you're building up large levels of any one given mutation that could be dominant negative and cause problems. However, it might provide cells the opportunity to make antibodies against a wider variety of peptides. The other thing to keep in mind is while these changes in translation induced by modification might not matter all the time. There might be certain situations in which they matter a lot, just like we saw with Chuan He's read-through data where those modifications didn't matter in most cell types, but they did in one. And those considerations, just things to think about, are things like tRNA pools, which certainly change between different cell types. And if you have a different pool of tRNAs around, you may be driving the kinetics of one misincorporation event forward. Also, the modification status of tRNAs in the tRNA pools can be quite cell type or stress dependent. And those things also are are also really important for translational fidelity and might be driving changes in translational fidelity, particularly when they're interacting with modified nucleosides. Lastly, different cell types contain different pools of RNA binding proteins, and these RNA binding proteins can be important for translational fidelity and might also be driving a sort of cell-specific effects of different mRNA modifications. So I would like to thank my funders, my wonderful collaborators, and my fantastic group, and thank you all so much for your time.
It's a great talk, so I'm sure Kristin would be happy to answer any questions.
Thank you, Kristin. That was very interesting. I'm a little confused about the molecular basis for how pseudo-U or N1-methyl pseudo-U can stabilize codon-anticodon interaction. So on one hand, you said by crystallography, the difference that you saw, or that they saw, was all the way at the amino acyl end of the tRNA, which suggests a long-range effect.
Yes.
And then,
So that's also with a cognate tRNA.
yeah, if you look at, like, so the interaction between pseudo-U and an A in a codon-anticodon interaction, it's exactly the same as the interaction with the U, and the extra N1 imino is not hydrogen bonded.
No, it's
and
not.
In fact, in all of the alternative base pairs that you showed, like, for example, a reverse Hoogsteen G pseudo-U interaction, you can make exactly that same interaction with U because the N1 imino is not hydrogen bonded. So in other words, there's still no satisfying molecular explanation for the stability at the basis of hydrogen bonding.
Right. So we have done a lot of other calculations that I didn't talk about here, where it changes the ring electronics quite a bit. And we think that that's really what's driving it. It has to do with the changes in the ring electronics are changing the hydrogen bonding patterns. And pseudo-uridine is well known to stabilize RNAs because it can form really strong hydrogen bonds with canonical bases.
Yeah, so I know that there's some like 20-year-old data where pseudo-U's was seen to give about an extra half kcal free energy, and it was never really explained, and the hand-wavy explanation at that point was that maybe it could coordinate a water molecule that could bridge to a phosphate. This is Kathleen Hall.
Yeah, I know exactly what you're talking about.
All that stuff, and then so my question is, since now you're doing some MD simulations, can you pinpoint that effect to, for example, changing the electronics of the Watson-Crick, or in a way that can provide a mechanistic explanation for the stability, because it hasn't really been put out there.
You're right. You're totally right.
Yeah.
So we definitely have done MD simulations looking at three base pairs together, but not in the context of the ribosome yet. And looking at the three base, it's like a codon-anticodon interaction fake thing. When we do that, it definitely changes the ring electronics. Without question, there are changes in the ring electronics. Some of it seems to be a little bit entropically driven, but there's a big enthalpy component as well. I can't explain it beyond saying I know that there are differences in the ring electronics, and that seems to be, or that's our hypothesis about what's driving it. If you have other ideas about what might be causing it, I'm very open.
It's a small effect, right?
Yeah, it's a tiny effect, which is why it's so shocking.
To the point where, it's hard to believe that you could recapitulate that with an MD simulation, with errors that would be believable, in my opinion.
Right.
There's something that is going on, that's clear. And I just haven't come across something that really pinpoints the molecular basis for the effect. But I think that changing the electronics of the
Yeah, and we looked at a whole bunch of different conformations because pseudo-U can supposedly do this like a syn-anti thing, but we could never make that actually fit in the ribosome. So we really think it's in the normal conformation in the ribosome.
One way people have stabilized interactions is to kind of pre-order the nucleotide so that it's in a conformation, like this is like what LNA lock
Yep.
nucleic
Yeah, yeah.
acids do, and those are way more stable because they have less dynamics in the single-stranded state. And so the entropy is already paid. So that would be another hypothesis, is that you're getting more enthalpy because the nucleotide, for some reason, is predisposed to be
Yeah,
in the
that's an
conformation.
interesting idea.
Time for
That's
maybe
something to
two
look
more
at.
questions. Great. Jim?
So, in your dipeptide experiments, are you using just a are you using just a triplet messenger RNA? I mean, a hexamer for the messenger
No,
RNA?
no, so it has UTRs. Yeah.
Hi, very interesting talk, Kristin. I noticed that when you were looking at the effect of the misincorporation, you saw no effect on serine, which is on a phenylalanine codon, which is a hydrophilic amino acid, and then the leucine and isoleucine had an effect in contrary. So I was wondering if you've done more comprehensive analysis on misincorporation on other amino acids and whether there would be any advantage in replacing a phenylalanine, which is nonpolar, with some other nonpolar amino acids.
We've wondered about this a lot.
Yeah.
We have not done the analysis yet, but that's something that we've wondered about a lot. Yeah. It's a good question.
And just as a follow-up, you were talking about the fact that this misincorporation or the pseudo-U is found under stress, right? Are you sure this is not just a consequence of the bad things that happen
So I
during
think it could
stress?
totally be a consequence, right? I don't disagree with that at all. I think that is definitely one possibility. It might not be a programmed thing at all. However, if you have a consequence that's happening on, say, 600 mRNAs, biology will probably take advantage of that.
I see. So your hypothesis is that it's actually a source of diversity, or there is some advantage
Yep.
for it, and it will be really interesting in the future
Exactly.
to study that. Thank you.
Don't think we'll keep Kristin any longer up here, but let's thank her once again for the wonderful talk.